All right, hi everyone. This is a lecture on the nervous system. The title is Neurons and Nervous Systems, chapter 37. Okay, so just like all these chapters, we will talk about some different types of nervous systems and some different types of animals. We'll talk about nervous tissue in general, then focus on the central nervous system and the peripheral nervous system of the human. All right. The intro to the chapter, right, talks about Michael J. Fox. So it says in 1991 that he was diagnosed with early onset Parkinson's disease. And what Parkinson's disease is a result of is something called degeneration of certain areas of the brain or certain neurons, meaning that when we say degeneration, there's neurons that are essentially dying. And so again, depending on where those neurons die, that's going to dictate what type of symptoms you have. And so for Parkinson's disease, it's in areas of the brain that are related to movement, but more sort of inhibition of movement and sort of regulation of movement, not just, thinking about like your motor cortex and just, saying I want to move my arm, but it's more of these uncontrolled movements. Okay. So that's why you see tremors, but also difficulty walking and speaking, again, because that is these are coordinated tasks that involve lots of brain areas. No cure. There is medication that can decrease some of those symptoms. But a lot of those medications also have a lot of unwanted side effects. There's an experimental procedure that I guess Michael J. Fox went through called a thalamotomy, which will basically, it's going to remove parts of the thalamus, I guess, and it helped relieve some of his symptoms, but they then returned. So right now, really the treatment for Parkinson's disease is to take a drug called levodopa, which is essentially going to replace a neurotransmitter that's normally produced by these neurons that are dying in the brain. And so it's just to try to bring up those neurotransmitter levels. However, too much of dopamine creates other unwanted, a lot of them are psychiatric type of side effects, like hallucinations, perhaps anxiety, depression. And so it's a tough disease because there's no way of specifically pinpointing those motor symptoms. Okay. And so anyway, he established a foundation to fund PD research. There's lots of Parkinson's disease type research that goes on. So hopefully we will come up with a better cure. I mean, not a better cure. I don't know why I keep saying that. I think it's because I see cure on the screen, a better treatment, right? For most diseases, we'll never find a cure. It's about developing proper treatments though. Okay. So in terms of some different types of nervous systems, right? We'll start talking about invertebrates. And these are invertebrates that we looked at in lab, invertebrates that we talked about in previous lectures. So Hydra, right? Remember had little living Hydra. We saw them in the dish and they sort of adhere to the bottom of the dish and kind of stand up. These guys have a nerve net. Okay, so all of the neurons in that nerve net are in contact with one another. And then they're also in contact with the contractile cells in the body wall, and that's what allows for movement of that Hydra. Planaria have a ladder-like nervous system. Okay, so there are two ventrally located bundles of nerves that extend from something called cerebral ganglia and then all the way down to their posterior end. And in the planaria, remember, we saw something called cephalization, and we talked about this before, meaning that you see this head region, okay? And it says here there's a concentration of ganglia and sensory receptors in the head. So ganglia are just cell bodies of the neurons that sort of associate or kind of are grouped together. And so we can think of that as being really the beginnings of a brain, right? So if you think about the brain, obviously the human brain, super complex, but lots and lots of neurons there, right? And so if you have a region, that's this head region where you see neurons sort of grouped together, that's the beginnings of a brain. You can kind of think of very, very simple sort of, organization of these cells, of these neurons that then would potentially evolve into an actual brain. Okay, so it says here bilateral symmetry and cephalization are two significant adaptive trends in nervous system development. And we talked about this too, like when we talk about human evolution, right? We talk about, I guess these are sort of kind of these two concepts, but like on a higher level, right? We talked about the development of a more complex brain and also the development of being a biped, right? And how that made humans more successful, or, hominids more successful than their predecessors, right? And so this is kind of like a more simple sort of concept, but along the same lines, okay? If we move on and we talk about annelids and arthropods, these are more complex, right? They have typical invertebrate nervous systems, and there's a brain, okay? So there's a brain present and a ventral nerve cord that has a ganglion in each segment, and we'll kind of see what that means, okay? But these are, again, more complex. Mollusks, they actually have a really well-defined brain and sense organs, and so octopi there's been studies that have shown that they can learn, and mollusks actually have a really complex nervous system, and can do more complex tasks than some of these other invertebrates that we talked about. Okay, so this is showing you here this nerve net of the hydra. Again, super simple, right? It's just kind of showing you neurons in contact with muscle cells in the outer, or contractile cells. I wouldn't even call them muscle cells. It's not muscle. In the outer wall, that are essentially these guys. These neurons would activate these contractile cells and allow movement in the hydra. Okay, here you can see planaria, a little more complex, right? You can see sort of this concentration of specific sort of cell bodies of neurons up here in this head region, which potentially then this will evolve into an actual brain, right? So like in the earthworm, you actually see a brain there, okay? Much more well developed. But then you can also see this sort of, so this is showing you this ventral nerve cord, if you kind of think of that as similar to like a spinal cord, but obviously way more simple, and then nerves extending out. Okay, so you can see how this is slightly more complex than this and more complex than this. And so you can kind of see that evolution occurring. And if we continue on, right, now we see a crab, right? Again, brain, and then this is showing you a specific thoracic ganglion. And remember, a ganglion is basically a collection of cell bodies of neurons in an area outside of the brain, okay? Here you can see for squid, right, we talked about mollusks of having really well-defined brain area, right? So it's showing you the brain here. You can see it's more well-developed, and then showing this sort of giant nerve fibers. And so actually the squid axon has been used and still is used a lot in understanding the action potential, right? And how the action potential is generated, and seeing it move down these large nerve fibers, okay? And these giant axons that they have. And then obviously if you move on to a cat, we talk about a mammal, much more well-developed brain. You can see the forebrain here, the cerebrum and the hindbrain, and then leading down to the spinal cord. Okay, so again, going from most simple to most complex. And then we'll talk about, obviously, the humans as being the most complex here, okay? So if we talk about the vertebrate nervous system just in general first, before we get to specifically talking about the humans, there's three main areas that you can organize that vertebrate brain into, okay? Hindbrain, midbrain, forebrain. So again, think about kind of location, right? If you think about sort of back of the head, middle of the head, and then the front forehead part of your brain, okay? So hindbrain regulates motor activity below the level of consciousness, okay? So if you think about the brainstem, part of the brainstem, the medulla oblongata, this controls breathing, controls heart centers, okay? So these are motor activities, involuntary motor activities, okay? So hindbrain or brainstem controls involuntary processes like breathing, okay? Like the heart beating. Midbrain, and these, again, this is sort of in general, okay? Midbrain, optic lobes are part of the midbrain, okay? So it says originally was center for coordinating reflexes involving the eyes and the ears. It has, more function, but again, this is sort of one of the main functions. And again, we're sort of talking about vertebrates in general and sort of simplifying this in these sort of three main regions. To talk about the forebrain, okay, it says early vertebrate evolution, forebrain was concerned mainly with the sense of smell. However, it has developed into much more than that, okay? Hypothalamus, I mentioned this in our Zoom meeting, and I'll mention it again when we maybe talk a little bit about that, but it's concerned with homeostasis. The hypothalamus controls the pituitary gland, controls the entire endocrine system, okay? that hypothalamus communicates with a lot of other brain regions as well. So pituitary gland I mentioned, but also with the brainstem, okay? All right, and so this is a picture showing you this. Again, I said to kind of think of hindbrain as being, you know, back of the brain or closest kind of towards the neck, right? So here's the brainstem, spinal cord, hindbrain, the cerebellum, optic lobe, which is part of the midbrain. Then we're talking about forebrain here being the cerebrum, the thalamus, and the hypothalamus, okay? So again, this is sort of a general organization of the vertebrate brain, okay? Then you're going to see there's lots of variation in terms of how well developed these sort of main areas are, okay? And then what other types of brain regions are even further developed within them, right? Okay, so now we can specifically focus on the mammalian nervous system. So hindbrain and midbrain of mammals are similar to those of other vertebrates, like we said here, okay? But it's really the forebrain that's greatly enlarged in mammals, okay? So if you want to, the outermost layer called the neocortex, or sometimes we just call it the cortex, it's only seen in mammals, okay? And that's where you're going to see functions of higher order mental processes like spatial reasoning, conscious thought, language, if you think about sort of the frontal lobe or the prefrontal cortex, right, which is this area that we're talking about, decision making, planning, personality, right? That's all kind of in that area, okay? It says here the frontal lobes are large and complex in primates, and we're going to look at diagrams, and I pointed this out in our Zoom meeting in terms of different brain regions. I'll do it again quickly, but hopefully we're sort of picturing what we went over a little bit in terms of where the frontal lobes are. So they're large and complex in primates. Again, that increased brain capacity allowed mammals, especially humans, to be able to perform these higher mental activities that you know, predecessors weren't able to do, okay? And then again, I kind of mentioned this in the beginning, but this is what gave humans a huge advantage compared to their predecessors, okay? This ability, or this complex development of the brain that gave them the ability to basically do these higher order mental activities, okay? All right, so let's talk a little bit more about that nervous system and how it's set up, right? So central nervous system refers to the brain and the spinal cord. Pretty simple. Peripheral nervous system, okay, is all of the nerves and the ganglia that lie outside of the brain and the spinal cord. Okay, now you divide that peripheral nervous system up into two divisions, somatic nervous system and autonomic. Okay, so somatic nervous system is sensory and motor function that controls skeletal muscle. So this is all sort of your conscious control, right? Moving your limbs, right? Feeling that something's hot, that type of thing, okay? Autonomic nervous system, again, is involuntary, right? Controls smooth muscle in your organs. You don't consciously control that, right? Your cardiac muscle, glands, okay? These are all things that you really don't, control on your own or are involuntary, okay? That autonomic nervous system then is divided into two more parts, sympathetic and parasympathetic, okay? Now, I put this here. This is an extra slide that I sort of add in. This is not from your book. And again, it's complex. It's not meant to show you or for you guys to memorize any of these parts at all. It's really meant to be illustrative, showing you sort of sympathetic versus parasympathetic. The idea is that sympathetic nervous system, right? You can think of sort of fight or flight in terms of its sort of function, right? It's going to increase things. It's going to increase heart rate, right? It's going to increase blood pressure, okay? And this is actually showing you kind of the setup of it and some of these sympathetic connections from the spinal cord and where they're going to in terms of their target organ, okay? But for the most part, when you think about sympathetic or an increase in sympathetic activity, it's increasing or it's speeding up, okay? If you think about parasympathetic, this is rest and digest. And it's going to innervate all of the, all of your organs as well, but it's going to activate the digestive tract so that you can digest your meal, but it's going to slow heart rate. It's going to slow your respiratory rate. It's going to lower blood pressure. Okay. So that's the kind of opposite. One is, again, rest and digest. Think of the sympathetic as fight or flight. Okay. So if we sort of just look at organization of the nervous system here, again, brain, right? Spinal cord. What's in yellow is peripheral nervous system. All of your nerves, right? And what's in those nerves are axons. So think about like lots of cell bodies of your neurons sitting in the spinal cord. Their axons extend out through those nerves, and then they can go and talk to these muscles out here, right? Or talk to a gland, okay? So a nerve is just a collection of axons, a whole bunch of axons within that nerve, okay, that are then encased by connective tissue or what have you. Remember we mentioned ganglion before, right? And you can kind of see some of these ganglion that are mentioned. So that just means here's a cell body of a neuron sitting here, okay? The axon extends out here, but then there's another neuron here. So another cell body that one will talk to, okay? So a ganglion is just a cell body that sits outside of the brain or the spinal cord. All right. So just to kind of show this in a schematic form here, central nervous system, brain and spinal cord, right? Showing you outputs going out, right? So this is motor. So somatic motor fibers, like I said, going to skeletal muscle fibers, right? Movement, okay? But then there's also autonomic motor fibers that are going to go to cardiac muscle, smooth muscle, glands, right? And this is all involuntary. And remember, that's the autonomic nervous system. You have a sympathetic division and a parasympathetic division, okay? If we talk about now information coming in, right? Sensory information coming in. So you have somatic sensory fibers, like things that are going to tell you about pressure, about, just touch, about temperature, things like that. That's going to come in here. There's also visceral sensory nervous sensory fibers, which these are coming from your internal organs. Okay, so these are things that you aren't really necessarily conscious of. Okay, like there'll be receptors within the blood vessels that would then tell the brain that you basically or the blood pressure is too high. So the smooth muscle in the blood vessel needs to relax to basically bring down blood pressure. So things like that you're not even conscious of. Okay, so like I said, we should know this organization of the nervous system, of the human nervous system. For sympathetic and parasympathetic, remember this is sort of fight or flight. This is the rest and digest part of it, okay? And again, when we talk about autonomic, this is involuntary, all right? Okay, so to talk about the actual tissue a little bit more, and we've already sort of mentioned this when we had our chapter on tissues, right? Actually, I mentioned probably all of this, right? That we have neurons, we have neuroglia. Neurons are the cells in the nervous system that can conduct nerve impulses, right? Generate action potentials. Neuroglia are those cells that help the neurons, right? And support the neurons, okay? Neurons, remember the three main parts. Again, we went over this before. Cell body, nucleus or organelles are in the cell body. Dendrites receive signals, okay? Axon is where that nerve impulse is going to be conducted down. And remember, the axon is covered by a myelin sheath. Okay, it says a long axon is also called a nerve fiber. Again, if you think of nerves, they're collections of axons, okay? All right, in terms of types of neuroglia, I don't know that we mentioned different types of neuroglia before, maybe not, okay? So astrocytes have a laundry list of functions. Here we're just gonna say they help maintain neuron health. Microglia are essentially immune cells of the nervous system. They're phagocytic cells, meaning they can remove bacteria and debris in the central nervous system. Remember, we kind of talked about white blood cells being able to phagocytose bacterial cells. Well, microglia are the immune cells or the white blood cells that do that in the nervous system. You have oligodendrocytes in the central nervous system that form myelin. In the peripheral nervous system, Schwann cells form the myelin sheath. Okay, remember that on the axon where you don't have myelin, it's called the node. These are called nodes of Ranvier, right? So here, where you don't have the myelin, we saw this picture before, okay? Covered by myelin sheath, but you have spots where there are nodes or there's no myelin, and the action potential is going to jump from node to node, okay? All right. There are also other types of cells these are types of neuroglia. Ependymal cells, which will line the ventricles of the brain, and they actually produce cerebral spinal fluid, okay, or CSF. There's also satellite cells that can surround the neuron in the ganglia. So these are outside of the central nervous system, but in the peripheral nervous system, and they're involved in responses to injury. So these are examples of different types of neuroglia. I could definitely ask you to describe some of these functions on the final exam. Okay, again, reminder, make sure you have your review sheet and you are taking notes on your review sheet as we're moving through. Different types of neurons, right? There are motor neurons, or efferent. Think of efferent exit. Motor neuron, basically they're going to transmit impulses out, right? Send information out. So it says they transmit nerve impulses from the CNS to muscles or glands, okay? In general, we can call these multipolar. Don't worry about that, to be honest. Let's just know the function of these different types of neurons. Sensory or afferent neurons. Again, this is taking information into the central nervous system, right? So it accepts impulses from sensory receptors. Again, like a sensory receptor on your finger, right? Underneath the skin of the finger, detecting whether something's hot. That information is then going to get transmitted into the central nervous system through sensory neurons, okay? Then there are also many, many, many interneurons that basically convey information or convey nerve impulses between different parts of the central nervous system. So when we go over the brain parts, think about all those brain parts that we talk about. They need to be able to talk to each other, okay? And it's through these interneurons. So, right, it says they form complex pathways in the brain. Lead to higher mental functions, thinking, memory, language. This is all due to these complex networks that are formed between brain regions within the brain. And the networks are basically composed of neurons, okay? All right, and so again, we've seen this before. We should know the parts of the neuron. All right, so this is basically just showing you, I said kind of don't worry about the different types of neurons, or don't worry about having to identify a unipolar versus a multipolar neuron, but just as an illustrative sort of purpose here, this is showing the sensory neuron. The reason why we're saying it's unipolar is because sort of this is just one process here, this axon, and it's split, okay? Like I said, you guys aren't going to have to worry about these details, but you can see, again, think of sensory receptors in the skin. Like I said, that might detect temperature or pain or something like that. And then that information is just going to go in, okay, to essentially kind of go through this sensory neuron and eventually going to end up here at the axon terminal, and then would talk to another neuron in the brain, right? Or in the spinal cord. Okay. If you talk about interneurons, you can see here they have a whole bunch of dendrites here, right? And then just one single axon, meaning though, we said that interneurons are involved in forming these complex networks in the brain, right? And so think about this kind of where each one of these, and this is even oversimplifying, where each one of these dendrites ends, there's a neuron talking to this one single one, right? And this neuron has to be able to integrate all of this information coming in, right? And then make some response and then talk to its targets over here, okay? So you can see how complex, and again, this is way, way oversimplified, this sort of the nervous system is. Okay, now, I mentioned a couple times transmission of nerve impulses, right? And I probably mentioned this before. If we're talking about, again, if you look at this picture, and this is, again, the easiest to conceptualize, a motor neuron, right? That's part of the somatic nervous system. That's going to stimulate a muscle, right? And tell it to contract. So if you think about, you just kind of hold your arm up, your palm facing up towards the ceiling. If you're going to lift your hand towards your face, your bicep is going to contract, okay? So in order to do that movement, there needs to be neurons that are stimulating those muscle cells within that bicep muscle, okay? And so they do that by sending a nerve impulse or an action potential down that axon that gets to the terminals, and then essentially there's going to be a neurotransmitter that's released at these terminals that will then go and bind to the muscle cells and activate them, right? And basically generate an action potential there and tell those muscle cells to contract, okay? So that's what this next part is talking about, specifically the transmission of that nerve impulse down that axon, okay? So you have to understand a couple of things. So membrane potential, understand that every cell membrane has a certain membrane potential, okay? And that just means it's a difference in charge across the membrane, okay? And when you talk about an action potential, or the nerve impulse moving down. Sorry, I'll go back here. This nerve impulse moving down this axon, it really all it is a change in charge moving down, or change in that membrane potential, okay? And so again, I try not to get into too much detail here, but I do want to just hit a few main points about this. Now, okay, so that membrane potential in general is just a general term in terms of describing the charge across the membrane, okay? And that's what this is saying, electrical potential difference, or difference in the charge across the membrane. Now, all neurons and all cells for that matter have a certain resting membrane potential, or resting potential. Okay, so this is when the axon's not conducting an impulse, it's at rest. The inside of the neuron is about -70, or we say the membrane potential, or the resting membrane potential of the neuron is about -70 millivolts, okay? And it's always describing the charge inside, on the inside of the membrane versus the outside, okay? It says due to, in part, the activity of the sodium-potassium pump, yes. The resting membrane potential is maintained due to a lot, it says in part because there's other types of channels that are involved too, but a lot of it is due to the sodium-potassium pump. Don't worry about that, okay, right now. Action potential, what is it actually? And I kind of just said this. It's basically, it says a rapid change in polarity, a rapid change in charge that basically across the membrane, okay? It says an action potential is generated only after a stimulus larger than threshold, okay? So there are times when a neuron might encounter a stimulus that might cause their membrane potential to change slightly, but if it doesn't reach threshold, then an action potential would not be generated, okay? Now, there are certain ion channels within the membrane of the axon that are responsible for the generation of this action potential, okay? So it says here, one channel protein suddenly allows sodium to enter, and another will allow potassium to leave. So bottom line is, sodium and potassium gated channels are involved in the action potential. Again, you guys don't need to know details of what's actually happening. I just want you to know some of these main terms, okay, and what they are. So here are a few other terms. So when the membrane potential becomes more positive, right, that's the inside of the membrane becomes more positive. Okay, you can see here, right? This is showing you the inside. This is kind of weird here how this is. Anyway, this is the inside versus the outside. When this becomes more positive, we're going to call that depolarization, okay? What this is showing you right here, up here, maybe it's better to look at it this way. And I'll come back to that other slide in a second where we had the terms. But one, this is showing you resting membrane potential. So you can see here, this is the inside of the membrane, okay? And it's negative, okay? -70 refers to this charge on the inside. Now, once the membrane starts depolarizing, which just means it becomes more positive, okay? You see this here? Some of these negatives have turned positive. It means that this charge in the inside of that membrane is becoming more positive. So it was -70, now it's -55. What is this due to? It's actually due to sodium ions flowing into the cell. Don't worry about the ion channels and which ones are opening when this is happening, okay? That's for another time. You guys don't need to worry about it right now. But if you're interested, it is telling you what's happening here. So I'll reiterate. The action potential really is just a change in charge across the membrane. How does that charge change, or what's responsible for that change in charge? Has to do with ions moving, and particularly sodium ions and potassium ions, okay? So that's what's causing the change. If positive ions are allowed to move into the cell, it's just going to make it more positive. Just think of it that way, okay? So depolarization is when the membrane becomes more positive, all right? Here it says repolarization. This is when the membrane potential will return back to resting potential. So now it's becoming more negative. So you can see here, okay? So again, now it's getting even more positive in this picture, right? +35, okay? So it's kind of getting to its peak here at +35. That's what this is representing here, because lots of sodium ions flew into the cell, or were able to flow into the cell, and it became more positive. So this is showing you a graphical representation of the action potential, right? your membrane potential started off at about -70, right, which is resting. It started to depolarize and get more positive. Once it passed this point threshold, the action potential was then generated. Okay, this is again called depolarization. It reaches a certain peak here, and then it starts to repolarize, meaning the membrane's potential starts becoming more negative again. It sort of gets a little bit below resting, but then comes right back to resting. Okay, so this is showing you an action potential. What's actually happening here is a change in membrane charge. Okay, so this is showing you voltage across the membrane over time. So we should know, like, if I showed you this graph and asked you what is this phase versus this phase, we should know depolarization versus repolarization. Depolarization, the membrane becomes more positive. Repolarization is becoming more negative or getting closer to resting, okay? The depolarization phase has to do with or is due to sodium moving inside the cell. The repolarization is due to potassium moving outside, okay? But I really want you to just be able to identify the parts here in terms of depolarization versus repolarization, be able to recognize that this is showing you an action potential, okay? Also be able to label resting membrane potential on here. In terms of where the ions and which way they're moving, you don't have to worry about that. All right. Okay, now what I just showed you was kind of how that action potential is generated. Well, this propagation part is talking about how it moves along that axon, right? So I mentioned this already before, that it jumps. So I got to go back again. Sorry, I probably should just put this picture in again. It jumps from node to node. So where there's no myelin, action potential generated, and that's how it moves. Okay, and that's why myelin makes the, well, one, it insulates the axon so that here you have a faster conduction of the action potential, but because it's able, or a faster sort of change in membrane potential, but because it's able to jump from node to node, it moves way faster, ridiculously, at a rate ridiculously faster than an axon that might be unmyelinated. And you do have unmyelinated axons in the nervous system, but that's a story for another time as well. Okay. So this part is talking about, well, how does it move? Okay, it jumps from node to node, and that's called saltatory conduction. Okay, it says conduction of a nerve impulse is an all or nothing event, right? Either an action potential is generated or it's not. There's no like small action potentials, okay? Or there's no big action potentials. It is or it isn't. And it also says here there's something called refractory period, meaning as soon as that action potential moves on, that previous section is not able to generate another action potential. So this basically ensures that the action potential moves in one direction, okay? Now, once that action potential, right, reaches, let me go back to my picture one more time, my sort of overview picture. Now, we just talked about kind of what's happening right at the membrane in terms of when the action potential is generated. We talked about it moving down the axon. Propagation is due to saltatory conduction. You get this jumping, okay? Well, now the action potential has reached the axon terminals. So what now happens? Neurotransmitter is going to be released into a space here right outside of the axon terminal, between the axon terminal and its target cell, which we're going to call a muscle cell, okay? So that's what point we're at right now. All right, and so that space is called a synapse. Neurons nearly touch, or it's where the space between the end of the neuron and the beginning of the muscle cell. Okay, so again, sorry, the whole thing is called a synapse. The actual gap is called the synaptic cleft, but think of the synapse as that whole area, the end of the end of one neuron and the beginning of the muscle cell, and the space in between, all of that together. Okay. The actual gap between is called the synaptic cleft. And again, it's saying between neurons, but it could be between a neuron and a muscle cell. So I like to use the neuron and the muscle cell example because it's easier to conceptualize. All right. So like I said, what gets released is a neurotransmitter. Okay. So the neurotransmitter gets released. I'm going to show you a picture. There's a lot of detail here to which you really don't need to worry about memorizing. Please don't. I'm going to show you in a, let's go back to the picture in a second, right? So what this is, what those words are describing is pretty much what's going on in this picture here. Here's your action potential. It reaches the terminal, like I said. What's going to happen is neurotransmitter is going to be released. And you can see the neurotransmitter is actually packaged in a vesicle. Again, please do not memorize these details. If I'm going to ask you about this, I will include some sort of picture and ask you to interpret it, okay? So again, this is why I said you're not going to really be able to Google your answers on the final exam, okay? It'll be questions like this. Look at this diagram, and I'll give you a specific question relating to that diagram to explain some process, okay? So that neurotransmitter gets released. So again, let's think about this part, and this as being a muscle cell, and this is being a neuron, right? Or it could be another neuron, right? If we talk about these complex networks and basically different brain regions have to be able to talk to one another, well, they do it this way, right? So once the neurotransmitter is released into this space, the synaptic cleft, it will go and it will bind to its receptors on its target cell, okay? And that target cell, like I said, think of it as being a muscle. Once this binds, it will cause that muscle to contract, okay? And so what are these things that are released? Neurotransmitters. So the movement of that nerve impulse down the axon, or that sort of communication down the axon, is electrical, right? Once though it reaches the terminal, now it becomes chemical transmission, right? A neurotransmitter is released. So what are some types of neurotransmitters? Acetylcholine, right? Norepinephrine, dopamine, serotonin. These are all examples of neurotransmitters, okay? Acetylcholine is the neurotransmitter that is released at the neuromuscular junction. So if we talk about skeletal muscles contracting, it's acetylcholine that's going to bind there, okay? So it says it excites skeletal, but it actually inhibits cardiac muscle. All right, fine. Norepinephrine. So we should know and again, this is just a snippet of neurotransmitters. There's many others. We should know the sort of basic function of each one of these neurotransmitters, okay? So norepinephrine, importance as to dreaming, waking, and mood, but it's also involved in control of the autonomic nervous system as well. Dopamine, emotions, learning, attention. I mentioned dopamine in the introduction to the chapter, if we remember, related to Parkinson's disease, okay? So it's also involved in movement and sort of control of sort of unwanted movements. But like I said, if you take drugs that will increase dopamine, it leads to sometimes psychiatric issues because dopamine is involved in the control of emotions, also involved in learning and attention, okay? Serotonin, it says involved in thermoregulation, sleeping, emotions, and perception. We think about serotonin as sort of the feel-good neurotransmitter, okay? So we should know basic functions of these 4 neurotransmitters, and we'll leave it at that. There are many more. All right. Now, the one other thing to kind of mention here is I said, the neurotransmitter will bind to its receptor on that cell on the other side, right? That postsynaptic cell. But it doesn't hang out there forever, okay? So there are mechanisms to remove that neurotransmitter, because if it kept hanging out here, it would basically keep stimulating that cell. So think about that as being a muscle. It would just keep causing it to contract and it would never be able to relax. So those neurotransmitters have to be able to go away and only be released when necessary. So there can be enzymes like acetylcholinesterase that will break down that neurotransmitter, right? So I gave you the example of acetylcholine being released at the neuromuscular junction. Like I said, if acetylcholine hangs out there, it's going to keep stimulating that muscle, and that muscle will never be able to relax. It would stay contracted, okay? So there are enzymes that will degrade or break down that acetylcholine to remove it, okay? And then if the body or if your nervous system wants to contract that muscle again, well, it's going to have to send a new signal, okay? And that's what this is saying here. It says short existence of neurotransmitters at that synapse prevents continuous stimulation, like I said. There are also types of neurotransmitters that will inhibit or slow things down or cause another neuron to be slowed down or deactivated. And so again, if that neurotransmitter hung out there, it would keep doing that, okay? That's not good, right? The nervous system is constantly or needs to constantly be able to regulate and change things, okay? So those neurotransmitters need to be removed from that synaptic cleft pretty rapidly. All right. It says here synaptic integration, and I kind of mentioned this already. It says a single neuron is on the receiving end of many excitatory signals or many inhibitory signals. So again, what do we mean by excitatory and inhibitory? Excitatory meaning these are signals that are going to generate an action potential. They're going to excite the neuron or they're going to cause a muscle contraction. Inhibitory are signals that are going to make it less likely that an action potential would happen, right? They're going to turn off things, okay? So if I go back to where I gave you this, kind of showed you this before, I said here, right? This neuron's receiving many different inputs, and then based on whatever inputs it's receiving, it sort of integrates them and processes them, and then generates some response based on all of these inputs, okay? And so that's what this integration is that it's mentioning here, okay? It's the summing of all of those inputs, right? And so this is showing you in another way. This is showing you an electron micrograph where you can actually see this is a cell body in orange of a certain neuron, and then these are all those inputs coming in, okay? And so you can see it's got to integrate all of these signals, okay? And so that's what this integration is. So some of the signals may be excitatory, some are inhibitory. Well, kind of the integration and that response that is generated is a combination of all of these, okay? And it's sort of additive. So if there's more excitatory signals, then it's going to cause an action potential. If there are more inhibitory, then it's not, okay? So you can see in this case, if you look at this integration here, there was never an action potential generated, right? Because it never reached the threshold, okay? Again, I'm not going to ask you guys about specifics here, okay? So please don't spend too much time here. All right. In terms of some parts of the central nervous system, okay, getting over some parts of the brain and then talking a little bit about the spinal cord, and I think we end talking about spinal reflexes, okay? 3 specific functions, okay? So what we were talking about before was really focusing on the tissue itself and actually the cells, right? What's happening on the cellular level. Here now we're getting more bigger picture, okay? So 3 specific functions of the central nervous system: receives sensory input, then performs integration and generates some sort of motor output. Okay, pretty simple. Not really, right? Okay, we already said this: central nervous system, brain, and spinal cord. Spinal cord and the brain are wrapped in 3 protective membranes that are collectively called the meninges. And I mentioned this when I went over the parts of the brain. And then the spaces between those meninges is filled with cerebral spinal fluid, or CSF. That cerebral spinal fluid also goes through the ventricles of the brain as well, okay? As I said, that fluid cushions and protects that central nervous system. I'm sure we've all heard of meningitis before, okay? Whenever you see itis, it means inflammation. So meningitis is inflammation of the meninges, okay? And so this is caused by either bacteria or viruses that invade those meninges, and actually determine if you have meningitis or what type of meningitis you have. You know, a clinician will have to go into sort of the base or into your vertebral column, but where the spinal cord sort of ends there, and go in and pull out cerebral spinal fluid to be able to analyze it, okay? All right, so that brings us to the spinal cord. 2 main functions of the spinal cord: reflexes, reflex actions, and we'll go over an example of a reflex arc. There's a lot more, again, to say about all of this stuff. This is just overview. Means of communication between the brain and the spinal nerves, okay? Composed of gray matter and white matter. So when you talk about gray matter, that means that is part of that's the nervous tissue that has cell bodies. When you call something white matter, there's axons there, okay? And they're myelinated. That's what gives it its white color. All right, that's what it's saying here. Okay, so let's talk about the parts of the brain. So again, you are responsible for identifying the parts of the brain in red in these 2 figures here on that quiz that I'm going to give you. That's basically the brain anatomy quiz. On the final exam, I am not going to put pictures and ask you to identify parts of the brain from the pictures. We're doing that in the quiz, but for the final exam, we should know basic functions of these brain areas that we're going to talk about here, okay? So cerebrum, largest portion of the brain in humans. It communicates with, coordinates activities of the other parts of the brain. If you look at that cerebrum, there is a longitudinal fissure that will divide that cerebrum into right and left halves. So that's this here. This is the longitudinal fissure, okay? 2 cerebral hemispheres are connected by tracts within the corpus callosum. Okay, so if you look, if you look here, this is showing you the corpus callosum. Now remember, this view is basically this cut in half and then looking towards the inside there, okay? So this is the middle of the brain, and then what you can't see on the other end is the, you know, either the right or the left side. Remember, equal right and left halves, okay? So the corpus callosum is basically white matter tract that connects the right and the left hemispheres, okay? If you look at the surface of the brain, it says there are shallow grooves called sulci, okay, that divide each hemisphere into paired lobes, okay? And so that's when I was talking about these different lobes of the brain. So here's central sulcus, that's just one. You can see again on the surface of the brain, you have lots of gyri, these valleys, and then ridges, the sulci, okay? That's the surface of the brain, of the human brain, is not smooth, okay? It has all of these ridges and valleys, okay? So these are sulci are the grooves, and the gyri are the raised parts, okay? So in terms of the lobes, I told you are responsible for being able to identify them, okay? So if I switch here now, we look at the frontal lobe, right? Okay, parietal lobe, the occipital lobe here, and then temporal lobe is here. Again, you were responsible for being able to identify everything in red. Okay, so the primary motor area sits right in front of, or anterior of, the central sulcus, right? Okay, that's in blue. The parietal lobe is this whole area here, primary somatosensory area, sits right behind or posterior of that central sulcus, okay? Occipital lobe and temporal lobe were responsible for identifying, okay? So that's here. Now, if we talk a little bit more about that cerebral cortex, okay, I already said this. Okay, so I already said this stuff, right? Contains motor areas and sensory areas. I drew your attention to the primary motor area here, right? And the primary somatosensory area, okay? Now let's talk a little bit more about some other parts of the nervous system. Now, I mentioned Parkinson's disease, right? At the beginning of the chapter, of that intro, okay? And so this basal nuclei area of the brain, or called striatum, right, is an area that's involved in integrating motor commands, okay? It says ensures that the proper muscle groups are either activated or inhibited, right? So I said kind of when you think about Parkinson's disease, it's a sort of error in suppressing unwanted movements, right? And that's why you have tremors, okay? And then you have other motor issues as well, okay? And so again, it's results of loss of dopamine producing cells in the basal nuclei, okay? And so I mentioned this again at the beginning of class, okay? Or beginning of the lecture. So that's one area not identified on this diagram, so don't worry about identifying it, but we should know function. And again, the reason why we're pointing that out is because I mentioned Parkinson's disease in the intro, okay? In terms of other parts of the brain, now just don't, it's drawing your attention to saying diencephalon. Let's just talk about the sort of primary, the actual brain part, okay? So hypothalamus, involved in maintaining homeostasis, controls the pituitary gland, okay? And so if we go back and we look at where that hypothalamus is, it's right here, okay? And you can see where the pituitary gland is. The pituitary gland hangs right off the hypothalamus. So again, what we're focusing on knowing now is the main function of these brain areas that I'm going through, okay? All right. For thalamus, okay? Again, it's sort of right above the hypothalamus. It is sort of the relay station. So it receives all sensory input except for smell, and integrates that sensory information and then sends it to the cerebrum, okay? Pineal gland, you don't have to worry about identifying it from the picture, but if you look, the pineal gland is over here, okay? And it's involved in, so it's not nervous tissue, right? It's a gland, it's glandular. It secretes melatonin. So it's involved in maintaining normal sleep and wake, the normal sleep-wake cycle, okay? Cerebellum, right? Kind of looks like a little mini brain sitting off the back here, okay? We should be able to identify that. What is the cerebellum involved in? A lot of things, actually. It says it receives sensory input from the eyes, the ears, the joints, and the muscles. Sends motor impulses out to the brainstem, to skeletal muscles. So one of the things about cerebellum, it's involved in balance. It's involved in coordinated movements, but it's also involved in learning and memory. It has a lot of functions. But if you thought about kind of one main function of the cerebellum, you might want to think about its sort of involvement in sort of motor things, okay? Balance, like I said, controlled or coordinated control, okay, of movement. Brainstem, right? Midbrain, pons, medulla. We should be able to identify those three parts from our diagram here, okay? Right? Midbrain, pons, medulla. These are all collectively part of the brainstem, okay? All right. And so midbrain acts as a relay station for tracks passing between the cerebrum and the spinal cord or the cerebellum, okay? And then pons, okay, is a bridge between the cerebellum and the rest of the central nervous system. So kind of both of these areas are sort of, I don't know, they're sort of relay stations as well. And you can kind of see where the midbrain, the pons, and even where the thalamus are located. You can see why they make sense that they're sort of relay stations, and certain inputs will come into certain areas, right? And then be processed and then go in various other areas. So it sort of depends on where the inputs are coming from. So like I said, for pons, this is, information sort of coming between, from the cerebellum will go through this pons and then to the rest of the central nervous system, okay, and vice versa, okay? So pons is kind of the relay between the cerebellum and the rest of the central nervous system. Midbrain, still things from the cerebellum might come through the midbrain as well, but also from the spinal cord and the cerebrum. So it sort of acts as a relay station as well. But it has other functions too. Don't worry about them. Medulla, reflex centers for vomiting, coughing, sneezing, hiccuping, swallowing. These are all involuntary sort of control centers, okay? We also said there's breathing centers there, okay? So that's the sort of function of medulla. All right. This particular activating system, you don't really have to worry about, okay? To be honest, don't worry about it. You don't have to identify anything from here. This is basically just sort of showing you this sort of system or network that exists, okay? That is involved in kind of alertness. Okay, so it says the reticular activating system arouses the cerebrum via the thalamus and causes a person to be alert. Okay, so when this system is activated, this is what causes that alertness. Okay, so just leave it at that. You don't need to know more about it. Limbic system. Okay, another part of the brain, and again, we're not able to see these brain regions from this view, and so that's why they're not pointed out here. So don't worry about that. But let's know about the limbic system, and just two main parts of the brain that are part of the limbic system: the hippocampus and the amygdala. Okay, so it says it incorporates whatever. Don't worry about that part. But function, integrates higher mental functions and primitive emotions. So when we think about the hippocampus, well, one, amygdala, fear. Okay, so you automatically should associate the amygdala with fear. Okay, the hippocampus is involved in learning and memory, things like spatial memory, but also like emotional memory. So see how it says kind of integrates higher mental functions and primitive emotions. The limbic system is involved in, associating emotion, okay, with memory. You can kind of think of that. So like, if there's a sound or there's a sight that, may trigger a certain memory. Okay, this involves the limbic system. So it's that association between emotion and learning and memory that happens there. And you can see why amygdala is involved, right? Fear, right? It's important, if you think about evolutionarily speaking, that something that's dangerous would evoke fear and that you would remember that, right? Because if you don't, you keep doing the same thing over and over again and you'll probably end up dying because you would put yourself in a dangerous situation. However, when that amygdala becomes overactive, it leads to fear when maybe you shouldn't have fear, right? And then leads to anxiety and potentially can lead to anxiety disorders, okay? So hippocampus and amygdala, closely associated parts of the limbic system, involved in, again, sort of emotional learning and memory, okay? And tying those things together. So again, like I said, you can't see these from that other picture there because it kind of sits in the sort of temporal lobe there. Okay, so you don't have to worry about identifying these from a diagram, but you can kind of see here's the hippocampus and here's the amygdala. The hippocampus actually has this sort of seahorse-like shape. All right. So continuing on again, the hippocampus again is involved in learning and memory. Okay, so we all know what memory is. Again, super important that we're able to remember things and that we're able to learn. Obviously, you guys probably realize that, okay? It says here, short-term memory is associated with the prefrontal area of the frontal lobe. So that, if you think about that frontal lobe, the very sort of front of it, the prefrontal cortex, which I don't know that we have it actually labeled here. Yeah, so here, see this prefrontal area? This is actually involved in like working memory and sort of short-term memory, but long-term memory is more associated with the hippocampus. And like I said, spatial memory, remembering where you are in space and being able to orient yourself in space has to do with the hippocampus, okay? So that's what this is saying here. So it says it gathers long-term memories and stores them stored in sensory association areas, makes them available to the frontal lobe, okay? But basically those long-term memories are stored in the hippocampus. Okay, so if you think about the limbic system, hippocampus and amygdala fear and emotion. Hippocampus relates fear and emotion with memory because the hippocampus is really primarily one of the main brain regions involved in learning and memory. There are many other brain regions that are also involved in learning memory as well, okay? But hippocampus, you can think of a hippocampus and amygdala as sort of working together in generating emotional memories, right? Or associating emotion with memory and with learning, okay? All right. The last part of this is the peripheral nervous system. Only a couple more slides, thank goodness, and we're almost done. So I had mentioned parasympathetic and sympathetic before, so we'll kind of come back to that in a second, or we come back to that again, okay? Remember, peripheral nervous system is everything that's outside of the brain and the spinal cord, okay? So if we remember we divide that peripheral nervous system into somatic and autonomic, okay? So we said this in the beginning of the lecture. Somatic system includes the somatic part of the peripheral nervous system, right, includes cranial nerves and spinal nerves, okay? And so these are nerves that are going to get information from, think about, again, if you want to think about easy, think about sensory receptors in your skin that are going to get information about pressure, about touch, about whether something's sharp or not, okay? And send that information in. Think about the ability to move your limbs, right? You're going to have those motor neurons basically saying, all right, I want to lift my hand up, so I got to contract my bicep, right? That was the example I gave you before. And so somatic system, again, voluntary. Autonomic, involuntary, two divisions, sympathetic and parasympathetic, okay? Again, the autonomic is going to control smooth muscles of your organs, right? Your blood vessels, cardiac muscles, and glands. And so this is basically showing you cranial nerves, right? So here's your brain. If you flip it over, this is looking at the underneath side of your brain. So if we did the sheep brain, we would be able to look at some of these nerves, particularly the optic nerve is easy to see, or this optic chiasm usually. And so obviously, where's that going? To the eyes, okay? You can see here the olfactory bulb, and you can see these other cranial nerves as well, okay? And so again, think about, this is sending information out. So in those nerves are axons. So the cell body sitting in the brain, and the axons are extending out to all the different parts of the body, okay? Cranial nerves obviously are going to go to, the face. They're going to go to the eyes. They're going to go to the nose. They're going to go to the face and the sort of head and neck area. When you talk about spinal nerves, those are then going to go, why did I, why? This is out of order, apparently. But let me go back. This part, remember this thing? That belongs, what I'm talking about now, just to kind of give you an example. It's just showing you, right? All of these nerves, these spinal nerves, part of the you can think about the spinal nerves are going to innervate all these different parts of the body. Okay. This is particularly showing you, again, the autonomic. All right. So, I mean, I touched on it kind of in the beginning, and then we're sort of coming back to it again. So maybe it belongs in both spots. But again, you don't need to know those details in any way. It's just meant to be illustrative. Okay. So getting back to that, right? So again, this is showing you nerves, cranial nerves. This is showing you spinal nerves, so nerves coming out of the spinal cord. And they're going to come out at various different levels, kind of like you saw on that diagram that I just showed you of the autonomic nervous system. And again, they're then going to innervate those different parts of the body. Okay, so a spinal nerve that's coming out of your cervical spine of your neck is not going to innervate or extend down to your toe, right? So it sort of anatomically makes sense where these nerves are located, all right? But a couple of things here. I guess, you know what, for this and for the, no, you know what, I guess on the final exam, I could show you this, right? I could also show you a picture here of the reflex arc and ask you to identify some parts from it, okay? For the brain region quiz, I'm just keeping it to those two diagrams that I talked about before in terms of identifying brain regions. But we should be able to identify different parts of the spinal cord. And we should know gray matter versus white matter, okay? Right? Gray matter versus white matter, right? And I mean, if I showed you this, we should know this is the vertebral column, right? That this is a spinal nerve, that these are cranial nerves, right? These are all things that we should be able to identify, okay? So just to give you an idea there, you don't need to distinguish between the ventral root and the dorsal root or anything like that. But let's be able to identify at this point that this is a spinal nerve, okay? This is a cross-section of the spinal cord showing you gray matter internally and then white matter on the outside. What makes up white matter? Those are axons, right? That are myelinated. Cell bodies make up the gray matter. So cell bodies are sitting in here, their axons are going to extend out and then maybe make their way all the way out to some part of the body, okay? All right. So like I said before, and I said this earlier, sympathetic versus parasympathetic, right? Think about sympathetic as fight or flight. Accelerates heartbeat, says dilates bronchi, like I said, increases blood pressure, okay? It's going to basically get those muscles ready to do something, right? To fight, to run away, okay? And so they're going to require lots of glucose and oxygen, so you're going to need to increase heart rate, right? To get that blood moving. Parasympathetic, housekeeper, again, relax or rest and digest, right? Promotes digestion, again, decreases heartbeat because this is when you're sitting down after a meal reading a book. This is what's kicking into play, okay? Know the difference between the two. All right, finally, lastly, let's talk about reflexes. Just very simple, just giving you an example. I don't know if anyone has somebody around that you want to try out your knee-jerk reflex. You basically hit the tendon. And you can actually look up a YouTube video for this if you want, very easily. You basically you can even take your hand or whatever, the pinky side of your hand and hit the tendon right underneath your kneecap, and you can see that reflex, okay? And so what composes a reflex or a reflex arc? Let's look at this picture here, okay? So, when we talk about a spinal reflex, what do we mean? We mean there is some sort of stimulus, okay, that information is going to be sent in through a sensory neuron, right? Okay, and it's going to go to the level of the spinal cord. This reflex, right, the response generated as a result of this reflex that information never goes up to the brain. Okay, so if we're calling it a spinal reflex, it's only going to the spinal cord. Why? It needs to be a quick, immediate response. If it went to the brain, it would take too long, okay? There'd be too much processing that goes on there, and that's unnecessary for this initial response, okay? Now, there's other parts of this sort of stimulus, or other parts of this sort of information that'll go to the brain. If we talk about like pain perception and that kind of stuff, well, that's going up to the brain. But if we're just talking about the result of being you moving your hand away from that nail that you just pressed your fingertip on, or that thumbtack or whatever, It's just going up to the level of the spinal cord. Okay, so this is basically showing you a sensory receptor in the skin. Again, think about you put your finger on a tack on a desk by mistake. What immediately happens? You lift your hand away. Okay, how does that happen? Sensory receptor in the skin detects that piercing, right, sharpness, sends it in through the dorsal root of the spinal cord, the dorsal root ganglion, right? You can see here now it's coming in. So here's your blue, your sensory neuron. It then sends the information to an interneuron here in purple. Then that transmits information to the motor neuron in orange. What is that motor neuron going to do? Stimulate that bicep to contract, to lift your hand away. You should be familiar with the parts of the reflex arc. I will put this picture on the final exam in some way and ask you about it. So if I got rid of some of these labels, I may say, you know, what is this? This purple cell here, what is it? An interneuron, sensory neuron, motor neuron. Okay, what's its function? What's it's doing? So understand what's going on here in terms of the reflex. All right, this just gives you a little, I think I pretty much went through all of this in the diagram. One maybe I didn't mention effector, right? In this sense, or in this case, the effector is this bicep muscle, right? Or these muscle cells within the bicep. Okay. Hopefully that wasn't too much. It was a lot. Like I said, you know, that's just scratching the surface of understanding the nervous system. I try to just keep it at an overview. It's hard not to get into those details. So I hope that you guys were taking notes in your review sheet, and I hope that you will ask questions and ask me to clarify some things if necessary in our class meeting, our next class meeting. Okay, thanks guys. Bye.